Hello and welcome to my channel. If you are new here, thank you so much for watching. And if you are a returning subscriber, thank you for your support. On my channel, I discuss everything medicine, from historical facts, unethical experiments in the past to drug trials that went wrong. So if you are interested, please don't forget to like, share, subscribe and leave a suggestion on the comment section below. In this video, I will be diving into one of the most horrifying drug trials that went wrong and has unfortunately led to death and permanent neurological damage on volunteers. So let's get started.
Medics rushed 6 research volunteers to this hospital in western France after they became violently ill while taking part in a drug study. Prosecutors have launched an investigation.
On January 7th, 2016, a drug trial started in Rennes, western France. 128 volunteers participated on the trial. 90 were given the drug and the rest given a placebo. The drug was administered orally to healthy volunteers as part of a Phase 1 clinical trial by BIA, a drug evaluation company based in Rennes, on behalf of a Portuguese drug manufacturer, BIA. The first symptoms appeared in one man on January 10th, 3 days after starting taking the drug. He was quickly hospitalized, and the others followed. The trial was halted the next day. The patients, all men, were ages 28 to 49. The head of the hospital's neurology department said that 3 men may have suffered irreversible brain damage based on magnetic resonance imaging scans. The drug is intended to help with mood, anxiety and motor problems linked to neurodegenerative diseases by having an effect on the endocannabinoid system, a set of brain receptors. BIA identified the drug as an inhibitor of an enzyme known as fatty acid amide hydrolase. Other fatty acid amide hydrolase inhibitors have been tested safely in Phase 1 and Phase 2 clinical trials. The BIA trial was the first to test BIA 1024-74 in humans. It was designed to explore whether the drug, which had already been tested in mice, dogs, monkeys and rats, is safe to use in people. Marisol Touraine, the Minister for Social Affairs, Health and Women's Rights, said in a statement that her office was informed Thursday evening about a serious accident that resulted in the hospitalization of the 6 men at the Centre Hospitalier Universitaire de Rennes, in eastern Brittany. BIA Pharmaceutical also said that it had followed all guidelines and regulations for clinical trials. The company, it said, is strongly committed to ensuring first of all the well-being of the participants in this trial and to determine thoroughly and exhaustively the causes which are at the origin of this situation. On December 15th, 2016, during a conference presentation by a scientist from the Portuguese company, BIA, it emerged that the firm did not use certain readings called pharmacodynamic data on how the drug BIA 1024-74 was acting in participants who had received a lower dose before taking the decision to increase the dosage. Helena Gama, head of BIA's Pharmacovigilance and Drug Safety Office, spoke for the company at the BPS meeting when asked why the company had not included the pharmacodynamic data in its dose escalation decision. She said, "The evaluation before starting a new drug escalation is based on safety evaluation and pharmacokinetic data referring to other data on how the drug is absorbed, distributed, metabolized and excreted by the body. We did not have any profile that precluded the path to the further dosage," she said. BIA was not legally required to use pharmacodynamic data, and its trial protocol had been signed off by France's medical regulator, the National Agency for the Safety of Medicines and Health Products. Catherine Hill, a biostatistician who previously served on the National Agency for the Safety of Medicines Scientific Advisory Board, is also surprised that BIA did not take pharmacodynamic data. "This seems incredible," she says. "Flying blind is a perfect image." Pharmacodynamic data can reveal when the copies of the enzyme in the brain are close to being saturated with the drug, a point at which levels of free-floating drug may rise steeply with increasing dose and potentially lead to unexpected off-target effects on other enzymes. The off-target effects can be species-dependent, which could explain why studies in rats and mice did not identify the drug's danger. Researchers used a technique called activity-based protein profiling, which allowed them to screen the molecule's activity against a very large group of enzymes in living human cells. They found that at higher concentrations, the bile drug disrupted the activity of several lipases, enzymes that break down fatty acids. Helena Gamma also released new data on 4 trial participants who survived but became ill. She said that they had not yet recovered from neurological symptoms including memory loss, headaches, motor disorders and tremors. Critics at the BPS meeting suspect that because the company was not including pharmacodynamic data in its decision to up the dose, it did not realize that fatty acid amide hydrolase was already saturated. The new study does not provide an explanation for the toxicity observed with the compound BIA 102474, but does highlight its promiscuous nature. The doses given to the volunteers in the group where the accident occurred were also unnecessarily high, as full inhibition of fatty acid amide hydrolase occurred at a much lower level. The long-term impact on the health of the more severely affected volunteers had not been clear previously, but the fact that they are still experiencing symptoms underlines the conclusion that the fatal reaction to the drug was not a one-off. That's the first time we know they are not well, that they are still damaged in some way. So there you have it, yet again, another medical trial that went wrong which led to death and permanent neurological damage on the surviving volunteers. If you enjoyed the video, please don't forget to like, share, subscribe and leave a comment down below.